Okay, welcome to the class, students. This is second last topic of this unit 15, living beings. It is structure of seed. In this slide, you can see many types of seeds, different types of seeds of plants. Seeds are of different types of structure according to their types of the plants. Okay, and at first, what is seed? At first, what is seed? A seed is a small embryonic plant which is enclosed in a covering called the seed coat. Outermost covering is called seed coat, and inside seed coat there is embryonic plant. Embryonic plant that is seed, and usually with some stored food. Inside seed there is stored food. Therefore, seeds are used as food. Then the formation of seed completes the process of reproduction in seeded plants. Okay, in plants, seed completes process of reproduction. From seeds, we produce new plants. Therefore, seed completes the process of reproduction. Then, why are seeds advantageous for plants? What are the importances of seeds to plants? As these are a few points. Seeds maintain dormancy until better environmental conditions arise. That means seeds get better environment, then new plants will develop, new plants will grow. Until then, seeds are in dormancy state. Dormancy state means that is very silent state, passive state, not active. Dormancy state means passive state. When suitable environment is obtained, suitable environment means good temperature, carbon dioxide gas, and water, then seeds germinate into new plants. That passive state is called dormancy. Maintain dormancy until better environmental conditions arise. When environment becomes good, seeds will develop into plants. Then another point, afford protection to young plant. Young plant means plant part inside seed. Okay, plant part inside seed that is protected at vulnerable developmental stage. Then before development of plant, uh, it is protected inside seed. Inside seed there is adequate food. Adequate food. Adequate means required food is there inside seed. Therefore, due to this food contained inside seed, photosynthesis is possible after germination. Before photosynthesis, seeds need food. That food is required, that food is obtained from inside the seed. Okay. Another important importance of seed is dispersal of plants. That means geographically from one place to another place, seeds are spreaded due to food contained inside seed. Different birds wants to eat seeds, and by carrying seeds, birds fly from one place to another. Then seed is transferred from one place to another, and in another place, from those seeds, new plants will develop. Therefore, Dispersal of plants is possible due to seeds. Okay, main importance are again dormancy state. That means inside seed there is passive state. When it gets a good environment, then it develops into new plant. Another protection. Protection means protection means uh, in plants seed is protected inside seed coat. Another inside seed there is food. Due to food uh, in germination of plants, that food is utilized by the seed, and after germination, after new plant is developed, photosynthesis will occur and the food is not necessary from inside. Another dispersal of seed, dispersal of plants. That means seeds are carried from one place to another by different birds and animals due to food content inside seed. Inside seed there is food. Due to food contained in the seed, animals and other birds wants to feed those seeds. In this aim, seeds are transferred from one place to another, and from that seeds, new plants will develop in different places. That is dispersal of plants. Dispersal means spread of plants from one place to another. Okay, then look here, seed structure. Mainly, what are the different parts inside seed? Externally, there are mainly two parts, seed coat. Seed coat means outermost covering. Small, thin layer is present outside the seed, that is seed coat. Then hilum. From outside, we can see small depression. Small depression. This depression structure, which we can see from outside, that is called hilum. Externally, we can see these parts: outer covering and uh, depressed part. This part, depressed part, that is hilum, we can see from outside. Okay, that is external parts. Then embryo means inside. Inside, what can we see? Inside there is cotyledon. There is one spelling mistake. Coty cotyledon, r, there is r, not r. Okay, n. Cotyledon, D-O-N. Okay, Cotyledon. Cotyledon. Cotyledon means two parts are seen in dicotyledons. This one part and another one part here is. Then cotyledon means two parts are there. Uh, Look here. Here is one. Here is one. One part is here. Uh, One part is here, and second. Sorry. Uh, One part is here. Okay. Whatever here. Uh, mm, okay. One part is here. Uh, One part is here. One part. Then second part is here. Two parts. Two parts are here. These are cotyledons. Okay, cotyledons. And in monocotyledon, there is only one part. These two parts are not there. Okay, we will study later on. epicotyle. Epicotyle means inside seed there we can see small another structure that is epicotyle. Plumule, P-L-U. There is also one spelling mistake, Plumule. Plumule means from the embryo when plants, seeds germinate, one part is developed in the form of leaves, another part is developed in the form of roots. Then the parts which is developed in the form of leaves, that is plumule, and the parts which is developed in the form of roots, that is radical. Okay, these are internal structure. Mainly cotyledons, plumule, and radical. These are few structure, internal structure of seed. Okay, then uh, okay, then what even? Okay, okay, uh, okay. Next slide. Uh, Look here. Seed coat. This outermost covering is called seed coat, and seed coats are different according to the types of seeds. Okay, according to the types, seed coats are of different thickness. Hilum, look here. In every seed, there is one depressed part that is called hilum. And from this hilum, seed is attached to the seed coat. Seed is attached to the seed coat, and here is embryo plant also. Embryo plant, that is parent plant. Okay, then embryo, look here. Inside seed, there is a small part from where new plants is developed. That is called embryo. Embryo is what forms the new plant. Yes. Inside seed, there is one part. From this part, new plant is developed. That part is called embryo. That part is called embryo. Now, cotyledon, look here. You can see here one example of maize monocot and another dicot seed structure. Look here. Cotyledon is the first leaf that germinates. Cotyledon, look, this one part, another part. In maize, only one part is here, no two parts. Only one part is here. This is monocot, and in dicot there are two parts, one left side, another right side. Okay, and in this cotyledon, food is stored. Food is stored in this cotyledon, and there is epicotyle. These two, if it is dicotyledones, then cotyledones are connected by one fleshy part, that is epicotyle. Epicotyle, okay. Mainly, cotyledone is important. Epicotyle is connection between two cotyledones in dicotyledones, and in monocotyledone, it is just below the endosperm. Endosperm means food, food content. Okay. Then in monocot there are only one part, one layer of endosperm. In dicotyledone there is two cotyledones. Okay. This is the structure of monocot and dicot. Okay, epicotyl is not so important for your grade. Okay, and plumule. Okay, plumule is the part from this embryo. From embryo, leafy structure will develop after germination. Inside seed, there is this part. before leaf structure is developed, and that part is called plumule. Okay, a shoot tip with a pair of miniature leaves. Small leaves-like structure is present inside seed that is called plumule. And from these radical roots will develop. Okay. Now radical. From the embryo, roots will develop. Then that part from where roots will develop is called a radical. The part of embryo from which roots will develop is called radical. Look here. Seed coat. Seed coat means it is also called testa. Seed coat, outermost covering, embryo, cotyledon, epicotyle, plumule, radical. Okay. Then you can see here this seed inside seed. This part, this bracket part is embryo. Here is embryo. Inside part is embryo. From between these two brackets, one red bracket, another black bracket. Yes. This is cotyledon, black bracket, cotyledons, and this red inside here there is embryo. Then from here a radical also will develop and plumule also develop. A radical is the part of embryo from which roots will develop. Plumule is the part of embryo from which seed will develop. This is general structure of seed. General structure. Okay. Okay. So till now, do you have any question or not? Do you have any question or not, students? No? Hello?
Hello?
Yes. Do you have any questions?
No, sir.
Okay. I'll revise then. Okay. Look here. Seed. Seed is the part of plant from which new embryonic plant will develop. Embryonic plant is enclosed inside seed coat, that is seed. Inside seed, food is stored. What is the function of seed? Seed completes the process of reproduction. That means new plants will develop. Then advantages of seeds. Advantages of seeds that is from here, mainly dormancy. Look here, dormancy until better environment conditions arise. Dormancy means passive state. From when suitable environment is obtained from seeds, new plants will develop. Before that, that is in dormancy state. Dormancy means passive. This dormancy means passive state. Passive state. Okay. Dormancy means passive state. Another importance is protection of young plant. Protection of young plant in the form of seed. Protection of young plant in the form of seed. It is also importance of seed. Another is adequate food supply. Adequate food supply to the plant until photosynthesis is possible. When seed is germinated, it develops into a small baby plant. Then cotyledon will collapse. Inside seed, cotyledon will collapse. That will be lost, and there will be development of leaves, and then that plant can perform photosynthesis. Before that, adequate food supply uh, is the main importance of seed. food is supplied to the baby plant or embryo inside seed. And lastly, dispersal of plants from one place to another place. Seeds are dispersed, spreaded by animals, plants, and by winds also, by water also. And then what happens? Those seeds will be carried to different places, and in those different places those plants, those seeds will germinate into new plants when they get suitable temperature and water. Then then new plants will develop there. Therefore, for dispersal of plants also, seeds are important. Okay, these four: dormancy until better environmental conditions, protection of young plant, adequate food supply for the embryo, and dispersal of plants. These are importances of seeds. Okay, then uh, mainly in seeds, there are two parts, one external part and another is uh, internal part. External part is this one, seed coat and hilum. Seed coat is outermost layer, outermost covering of seed. Hilum is small depression in seed, small depression in seed, small this. This is hilum, and seed coat is this outermost covering. Outermost covering is seed coat. Okay, then mm, then another is internal structure. Internal means that is embryo. In embryo, cotyledon is present. You can see here two cotyledons are shown here. uh, Two cotyledons are shown here, one this and another this. Two cotyledons are shown. And epicotyle means these cotyledons are connected internally. Here is connection in dicotyledonous. Okay, that is epicotyle. And plumule means disease. Leaf-like structure will develop after germination. Then that is plumule. And radical is the part from where it will develop into root, that is radical. In this way, external part and internal part are there. They are external is seed coat and hilum, embryo is internal. Okay, in this way, uh, these are few structure of seeds. Okay, then seed coat protects the embryo. Inside seed coat, there is embryo. Embryo is protected by seed coat. Okay, and according to the types of the seeds, seed coats are of different thickness. Some are very thin layer, some are very thick layer. Okay, then for example, walnut. Walnut has very thick seed coat. Walnut, very thick seed coat. And other bean types, soybean. Soybean have very thin layer of seed coat. That is, according to the seed type, seed coats are also different in thickness. Okay. Now hilum. Hilum is a small depression which we can see from outside. That is hilum, small depression. Okay. And in this hilum, seed coat is connected to the embryo. Seed coat is connected with the embryo. That embryo is called parent plant. Okay, parent plant and embryo. Parent plant means parent plant means embryo. Okay, parent plant means embryo. Okay, uh, okay, then look here. Embryo is inside seed. And inside seed there is embryo. Embryo have two parts. One part it will develop leafy structure, shoot system. Leafy structure will develop after germination. Then that is plumule, and from down part roots will develop. Then that part is called radicle. The parts of embryo from which leaves will develop is called radical, and the part of embryo from which roots will develop, that is called radical. Okay, radical and plumule. Okay, then cotyledons. In seeds there are two parts. One is in monocotyledons there is only one layer of seed, that is called monocotyledons. And in dicotyledonous plants, seeds have two layers of cotyledons. Then these are cotyledons. Cotyledons have food content. Cotyledon have food content, that is called endosperm. Endosperm means food content in the food, in the seed. Outermost layer is seed coat, and internal part is mainly endosperm, that means cotyledonous part. And between these two cotyledons, in dicotyledons, there is one epicotyle. And in monocots, just below the endosperm, there is one soft fleshy part, that is called epicotyle. Okay, look. What is plumule? The part of embryo from which new leaves will develop after germination is called plumule. What is radical? The part of plant from which new roots will develop is called radical. The part of embryo from which new roots will develop is called radical. You can see here same part again revised. Seed coat is outermost layer. Embryo is internal layer. And there is cotyledons, epicotyle, plumule, radical. These are when we dissect one seed, when we cut one seed, what are the materials required for that is soaked into seed. Seed is soaked into water. Now that is very soft. Then we cut by dissecting microscope. There is one dissecting microscope to observe those parts. And there is tweezers and rulers. Yes, these are tools to observe seed structure. Okay. Okay, students. Is it clear now?